Good morning, everyone. Welcome to I think this is the fourth lecture of Martin. So today we will conclude the round of the fourth lectures. This afternoon it will be again Ilya's turn. And Martin, we are really very keen to see how the genomic story continues. And just one logistic thing. Martin, at around 10, I need to peek into another meeting of some Academy of Sciences of sorts. So I might ask Yazira and Ilya to moderate the questions at the end of your talk. Yeah. I will probably stay online, but be in another meeting. Yeah. So in case I don't finish before the end, I will ask Yazira and Ilya to take care of the end of the lecture. Yeah, if that is okay with you.
Sure, absolutely. Did I share the screen?
Not yet, Martin. At least I didn't see it yet.
Okay. Okay, let's see.
Yeah, now there is some action. Yeah.
Okay, so now I have to.
Yeah, now we see your brow. Okay, now we see your PDF. Yeah.
Okay, right.
Perfect. Wonderful. Nice.
Let's see, it's taking a little while. I think I have too many things open.
Oh, it's your new fiber, Martin. You need to warm up. Yeah.
Okay, so last time we talked a lot about biochemistry and technology, sequencing techniques. And I promised you in this lecture there would be more mathematical modeling. So we're going to talk, the sort of main theme is going to be mutations. I mean, there's a lot that is interesting without mutations taking place, but I mean, mutations were sort of the microphysical explanation to Charles Darwin's theory of evolution. And the early work was really limited toward things that you can see without knowing what the cause is or the molecular mechanism. So piece size, color, flower color, and stuff like that. If we talk about Mendel, we could talk, if we go to the beginning of the 20th century, we could talk about blood types and, compatibility of transfusions. So this was a, very important issue in medicine, being able to give transfusions safely. We'll talk a little about genetic diseases and what mathematical models tell us about explaining them. And I will also talk about molecular clocks, which will be important with establishing family trees for viruses and all sorts of organisms. And I'll focus more on that in the closing lecture. So I think I want to talk about the genetics of the coronavirus, and also from a sort of more mathematical point of view, what are the ways in which you can infer ancestry and relations from and differences between species, sort of a molecular fossil record just based on what you have now. So that's sort of a summary of the rest of the lectures. Okay, let me just back up a bit, and that these are some things that we details that we didn't go into in the last lecture. So I showed you the genetic code, how there are three successive bases on the DNA or RNA, so that give you 4 to the 3rd combinations. And so there are these words of 3 characters each that code with some redundancy the 20 amino acids. And then there were also some stop and start codons. So the codon is the 3-letter word. And, from that table, you would think that it would all be, very straightforward, just someone sequences, the human genome, some virus genome, or some plant genome, and you can figure out sort of what's going to happen from this computer program that you sort of know what the language is. So here the language has not been, it's not like Python, where it has evolved so that people can understand it. It just has to work. And one of the sort of we mentioned the complication of reading frames. So there's no, there are many ways to read a certain sequence, and you should think of it together with a complementary strand. It could be if DNA. On the strand that we don't call complementary, you could start, every 3rd letter, there are 3 ways to start, but then the protein could be on the other side. And there's ambiguity in how you read it. And so that's sort of one problem. And in bioinformatics, there are sort of tricks to figure out where what is probably the right reading frame, but they don't always work. And then where the transcription starts, there have to be promoters and that kind of thing. So it's, sequences sort of upstream. So it's not really possible to predict with great reliability. And in addition, there's some sort of editing that takes place, which is especially on vertebrate or more complicated organisms, where you have a gene, and there are these introns, and this all gets translated into a complementary piece of RNA, as you can see. And then before it gets translated into a protein, there's some editing that is done, and parts are deleted. So the protein is determined just by the sequence at the end after the editing. And that's sort of one of the problems to figure out where this cutting and pasting happens. So there, for example, with the Human Genome Project, there's some gaps, and it's not completely reliable, but it's pretty much a completed project. But people don't agree on what is the number of genes, because some of the things that sort of look like genes aren't. So you have to look at, in the cell, what is actually transcribed and translated. And that's sort of another big problem. So let's, this is a, timeline of the sort of sequencing technology. And on the next slide, we'll see something that looks a little like Moore's Law. The Human Genome Project, well, first, simpler organisms were sequenced, and proteins were sequenced in the 1950s, relatively short, 60 base residue length, 60 amino acid proteins. But there's been sort of an ever increasing amount of complexity possible. And the Human Genome Project, which had the ambition of completely mapping the human genome, that was started in 1990, and it was super expensive using the techniques then. But then there, as we discussed last time, there's the second generation sequencing is, much faster and less expensive. And I guess like mobile phones, there's new technology where the goal is to be able to sequence ever longer segments. So that project was completed. And then the other thing first you might ask, well, not everybody's the same. So what does that mean? Well, once you have sort of some average or reference genome, you want to know what are the differences. So there was this 1,000 Genomes project, which was completed. And the idea was to take sort of selection of people from all over the world, different, groups from all the continents who, haven't really been in touch with each other, and to map and to some extent characterize the full diversity. So this is obviously, very important for medicine, because the buzzword now is personalized medicine. So from looking at someone's individual differences from the reference genome, one in some instances now can predict which drugs will work and which won't, rather than having a doctor prescribe, and first you try this and first you try that. You take this data, and if you know the genomes of the patients, then you can establish correlations and you can predict right off what will work. So that's sort of one of the applications of this. And here you see, so this is sort of like a Moore's law plot. So except it sort of goes the other way. So what is the cost of sequencing a single human genome? So you can see that when this is in the middle of the Human Genome Project, we're talking about $100 million. So that's something that, you know, only very rich people like Elon Musk and Jeff Bezos would be able to afford. So it's a little like going out into space. And now, you know, $1,000 US dollars is, you know, not, it's expensive, but it's not exorbitant, especially in developing countries. And, you know, there's also a lot that can be done without sequencing everything. Okay, so I just included this plot, and I mean, we could talk for a very long time about the, you know, conclusions and what one has learned from the Human Genome Project. But here, in terms of information, we can see, you know, how many base pairs there are in the different chromosomes. So there are 23, and we have the 2 sex chromosomes, and they're sort of very roughly ordered in, you know, from the shortest, the most information, to the I mean the longest with the most information to the shortest. And the sex chromosomes will at the end maybe talk a bit about this. The male chromosome is much smaller than the female chromosome, and has, you know, if you just count in genes that code protein, it has less than 10%. So here you see there are about 20,000 genes that are believed to code protein. They're pseudogenes, or sort of genes that became defective. So they have a coding origin, but something went wrong. And then there's some other RNA that isn't translated, but it's also sort of a useful molecule to do different things. Only 2% approximately is actually codes protein, and there's a lot of other stuff. Okay, so let's look at, I mean, these are just tables to give you sort of orders of magnitude. So describing the differences in between genomes is a bit complicated, because there are different things that can happen. But most of the differences are just single nucleotide polymorphism, which means just one base changes. And you see that there's just, this is the number of kilobases per where there's a possible mutation. So it's 1 in 1,000 or a few thousand, you know, it depends on, it depends on the particular chromosome, and the male sex chromosome has the most. Okay, so that's not the only thing that can happen. There, sections can be added and taken out. So insertions or deletions. And when you make a comparison, it's a little ambiguous because, you know, they're, neither came first, so they're called indels. And a lot of mutations could be fatal. So if you just had one gene and you really need the protein and you delete 1 or 2 or not a multiple of 3 or add something that is not a multiple of 3, then you mess up the reading frame and you sort of get nonsense. So that's sort of a big problem, and that's not going to happen so often. But this is the table that you've already seen, and this redundancy is sort of interesting because there are some changes that they're called synonymous, especially in the third part of the codon where it doesn't make any difference. It might affect how often the protein gets translated or things like that. But that's sort of having the same code, a different code for exactly the same protein. And then missense is, you know, you just switch one and it's a different protein. And in the last lecture, we sort of saw that some proteins have similar properties. So it might not make that much of a difference and might not make any difference, or it could just completely mess up things. And we'll talk about comparing very differing organisms to sort of identify which are the important amino acids. Then if you put a sort of stop signal in the middle, that would completely mess up the gene. So that's sort of, these are the main types of mutations that can happen. And there are other sorts of differences in the non-coding parts of the genome. So these microsatellites are basically repeating units, so that you have a short pattern that repeats itself many, many times. And that is particularly error prone because, this can stick to this, but then you could get this to stick to this, and then you change the number. So there are a lot more frequent mutations, and there's really, within a certain range, the number of these satellites, there isn't really selection. So this is sort of, for example, what the police use to look at DNA matches, or these services, 23andMe, who you can find relatives and stuff like that. So that this is these repeating patterns mutate at a much faster rate. Okay, so the other thing about mutations, and this was very controversial for a while, is sort of using mutations as a molecular clock. So this was an idea of Linus Pauling and Emil Zuckerkandl in the 1950s, that there's sort of a constant rate of mutations among, different organisms, and over time. The people who thought that most mutations were had to do with fitness would sort of see genomics as, explaining these big changes, and that changes would happen in bursts. But here you can sort of see these divergences, and with fossil records, you can establish when you think the species diverged, and then look for consistency. And this seems to be like a pretty good fit. So here we will. This is a tree of species, and you see this kind of divergence. So that the horizontal extent is just so that things are separated, but this is the time axis. So if we just had one type of mutation, and it would be like radioactive decay, the number of if you would count the number of radioactive decays, you could make a clock. And for very short times, it wouldn't be very accurate between clicks of your Geiger counter. But if you had 1 million clicks of your Geiger counter, then you have a Poisson distribution. So the error would be, 1 over the square root of 1 million. So it's the accuracy of your clock is 1 part in 1,000. So that's the conceptual idea. So of course, we have much more confidence that our uranium decaying is, doesn't have these correlations, and it has the same half-life with time. And here it's only approximately true. But if you just look at this, if we have these genomes here, and we sort of want to test this hypothesis that this is the tree, there would be some non-trivial predictions that, for example, the genetic distance between this type of mouse and the rabbit would be the same as to this. And you can do it with all sorts of species, and how close is, I couldn't find this paper, but there's a lamprey or something like that, some very primitive non-vertebrate that is sort of like an eel, and how what is the distance between us and that organism in terms of certain proteins and, say a guinea pig, and you see that it's about the same. So you just sort of look at the distances, geometrically, because the angles are the same, and there are certain relations that have to be satisfied. Of course, the molecular clock could tick faster for certain organisms. There's no reason to believe that the mutation rate is, something like an atomic clock or something. But you can look at different genes and, get some internal consistency and calculate the distances in different ways. And that the phylogeny is basically, if you have all these genomes today, can you construct the tree? And there's also a probabilistic aspect to it, since mutations occur by chance, not because they have to, or by design, or that's the way we think about them. So there's sort of a notion of a most probable tree, and so this is one application of these molecular clocks. Okay. So I talked a bit about, I explained to you in general terms what mutations are and what diversity there is in, the human genome, and likewise any other species. And I just want to talk about a few examples of different alleles or different possibilities for a gene which have profound consequences. So sickle cell anemia is a big issue in Africa, not so much South Africa, but more equatorial Africa and other countries where there is a lot of malaria. So this is sort of one of the interesting connections. So we can understand, well, let me just show you the data, and very roughly, that you have, these are the countries where there's a lot of sickle cell anemia, and you can see that it's very much centered around the equator, and then this is a map of the malaria prevalence. So this is in 2009. There was malaria in parts of the US 100 years ago, Italy, Spain, Greece, but we're really interested in the regions where it is, very endemic. And the argument, which turns out to be correct, is that the sickle cell trait provides some sort of advantage to fighting off malaria. So let's sort of go to the, let's see, how does this, okay. So there are two, well, hemoglobin is made up of several proteins, but there is the normal protein that is one of the components, and then with a mutation, there's the sickle cell version. So what happens is the if there is both sorts of hemoglobin, the sickle cell, then the when the red blood cells get infected and have very little oxygen, they tend to burst. And that tends to be a protective mechanism against the malaria, because part of the malaria life cycle is to, after it gets out of the liver, is to infect and multiply in the red blood cells. So if the red blood cells will burst and not give much time for the parasite to multiply, that is a genetic advantage in terms of living in a place where there is a lot of malaria. But if both of the hemoglobin genes, so one from the father and the mother, have the sickle cell trait, then that leads to disease in these sort of defective blood cells that create all sorts of problems, in the absence of malaria, and really can shorten the life of someone who has both of these genes. So we could, let's see. So this is a sort of interesting situation in that the advantage is, well, if there's no malaria, there's no advantage to having the sickle cell trait. So you would think that if this causes disease, then you would get natural selection where that allele would be selected against and disappear, which explains that map, that if you ignore things like, migration, slaves being brought to the United States, etc., well, that explains why South Africa doesn't have very much sickle cell trait, but equatorial Africa does. But the natural selection will, where this is an advantage, so the external condition or boundary condition is what is the prevalence of malaria? Well, the heterozygous combination is a plus, but you can't have a population with this occurring all the time. With if everyone were heterozygous, then there would be 1 out of 4 chance of developing the disadvantageous trait. So this is sort of optimization condition where you want to have both alleles in a population, and not in a certain proportion, and not one is better than the other. So let's see, this is sort of the explanation with words. And here we can go to mathematics and do much the same thing. So here I take a generation. So we're going to have discrete generations in this model. You could make a continuous model, but that's just more complicated and we don't know the numbers. So what do? Well, we assume there's a large population and that people, when they choose their partners, don't know what their trait is, so it's completely random. So you get in the next generation what would be Hardy-Weinberg equilibrium. And then we apply these selection effects. So basically, this is the survival probability of the homozygous AA, the heterozygous AB, the homozygous BB, etc. And we're also going to renormalize. So the idea is that the total size of the population is determined by other consideration and can just be factored out. So this doesn't affect whether the population is expanding or contracting. That's sort of a side issue. And then we repeat this generation after generation. Let's see. So there are a few assumptions here. Well, let's see. We can just, because we renormalize, we can just set one of the selection coefficients equal to 1. Now, if we have a trait that where A completely dominates over B, then we just have one parameter. So this isn't going to be the sickle cell situation. And so the idea might be that, well, the gene A makes a protein that is needed for something, but one gene suffices, so there's hardly to make that protein, and it gets regulated, so it's not really observable or doesn't have any impact whether there is one or two gene. But when that isn't present, it's either a disadvantage or an advantage. So that's a bit of a special case. And then in the general case, the jargon is codominance. Well, all three coefficients, selection coefficients, would be the same. And then in the sickle cell trait, we can set the normal hemoglobin equal to 1, but we would want to have this be larger, so it's favored, and then this less than 1. And the assumptions, which are rather not always satisfied, is, I mean, we're analyzing one gene at a time. So there's random mating and there isn't a correlation. So it could be that, you know, in order for something to happen, you have to have protein 1 from one gene and protein 2 from another gene. So there are correlations, but that is a little not so simple to analyze. So here is a Python code that basically implements the algorithm that I just described and creates some data. So we look at different generations, and I will make this available. So the heterozygous is red, and the black is AA, and the blue is BB. And let's look at some parameters that would give something that could, what's a little unrealistic. So the starting condition is completely arbitrary, but here I give the heterozygous combination an advantage, and then the sickle cell trait, for example, I will assume that it's deadly, or that the people who have two sickle cell genes don't reproduce. So you look at what happens. So you first get this spike, and this is basically, this doesn't satisfy the Hardy-Weinberg equilibrium. So that in the absence of selection happens in one generation. I chose, these selection coefficients are a bit drastic, and I just adjusted them so that in 100 generations, or here a lot less, you approach an equilibrium. And you see that you get about 20% of the heterozygous genotype after some time, after an initial transient. So this is just the part that should be taken seriously, is that you can have a non-trivial fixed point where there is sort of a mixture of the two alleles, and if one dominates too much over the other, or vice versa, I mean, if there's a fluctuation about the perfect proportion, then it will get corrected. So you have a stable fixed point. But unless we really knew these selection coefficients, we wouldn't be able to predict this prevalence. And there would be this advantage in terms of selection would depend on how much malaria there is. And then this it's going to be lower than 1, but not necessarily 0. So that's also going to affect where the equilibrium is. Okay, here I chose something where there's a distinct advantage to the heterozygous combination, but there really isn't any significant disadvantage to having the two sickle cell alleles. And the reason why I made this 1,000 is so that these would separate a bit. So you can see that you have a non-trivial fixed point. Okay, let's do something else. So let's choose a case where we're going to have the B allele, the homozygous combination preferred. So we, here you get what I think would be described as a trivial fixed point. So in the end, B, everybody gets B after enough generations. In the A, allele gets lost because it's not as good in terms of fitness. So this is the kind of thing that would be sort of the mathematical illustration of Darwinism, how it works. And let's see, here we have selection against the dominant trait. So this is also trivial. I mean that one dominates over the other. So the dominant trait disappears. And let's see, where did I? Oh, this is mislabeled. It should be a recessive trait. And this is kind of interesting because here I changed this from 1.1 to 0.9, and you can see that the decay is much smaller. So the recessive trait allele is not good, but it only shows up where it's around quadratically in the homozygous combination. So there's, this is the rate at which the allele is disappearing, which is very slowly. So mixing in genetics and sexual reproduction tends to make recessive traits go down in prevalence, but then there's a very long tail. And that has a big effect in terms of diseases, because there are lots of these diseases where there's a not so common allele that hasn't disappeared. And this is from Wikipedia, all of the different autosomal is that it comes from the chromosomes that aren't the sex chromosomes, so that you have the heterozygous, homozygous distinction, but you don't see autosomal dominant disorders because those are selected against much faster. And the other, I just chose this for South Africa, but there are many examples of this, of populations which where there's a large group of people today who can trace themselves to a small founder population. So if people go to an island and they don't live there for many generations, then people can trace their genes back to the small population. And that can also be good, that it'll select, there will be recessive genes that have completely disappeared, but then they're, because no one from that small population had those rare recessive traits, that others will be a lot more prevalent. So this is an article about one of these diseases. Okay, so all these models are predictive in that they assume that the population is infinitely large, so there's no chance in terms of the number of the fraction of certain allele that is present. So if there's no selection, it would just stay constant forever. But let's go and look at the other extreme. So this is going to neutral theory, where there are different alleles that present themselves, but there's no selective advantage. So we're just going to assume a binary allele, and the population size will be n/2. Let's see, this should have been n. I sort of changed my mind where to put the 2. So if you want to figure out the number of alleles in the next generation, you have to draw from the binomial distribution. So we can do that, and then we get a random walk. But that doesn't mean that the random walk is going to fluctuate forever, because once everyone has that allele or nobody, then one of these 2 alleles gets fixed, or there's no variation, and the polymorphism disappears. So this is sort of one of the theories of, migrations, and, why people in different parts of the world have different characteristics, that you sort of have small groups of people that migrated, and in a small population, mutations can fix, and they don't need to have any advantage. It just happens by chance. So if you're sort of Darwinian, you could, and always want to see selection, you can come up with, elaborate explanations of why this and that is inevitable, but a complete random walk with non-infinite populations creates diversity. So what did I do here? I just coded this for 500, population of 1,000. And here you see that you start with allele that is very just 50, so 5%, and through the random walk, the fraction evolves, and then when it hits 1,000, the size of the population, it fixes, it sticks. So this is not so likely to go up like this. It can happen. It's more likely to hit the boundary that is is closer, but so most mutations that occur will never acquire a large fraction, but some can wander to 1 to taking over. So this is the sort of other extreme, that there are mutations that absolutely don't matter, but due to a finite population size, they fix, and you will get changes that are sort of inconsequential. And this is very good for the molecular clock theory, that you have these changes that sort of happen independently rather than being part of any great scheme. Now, nobody really seriously thinks that it's either one or the other. Okay, so was more and more data available, people realized that there were many mutations to get rid of bad mutations through selection. So this is where this neutral theory of molecular evolution came from. So it's a Japanese geneticist, Moto Kimura, who came up with this theory, and not everybody liked it, and there was a lot of controversy, and there's a lot of discussion in the literature to what extent mutations are generally neutral. So he doesn't claim that this always happens, but that this is the case most of the time. So I basically combined the two programs. So I took the natural selection program, and I put a finite population in, and then rather than taking the P and feeding it here, this is the allele probability, I sort of do a binomial draw here, and you have these parameters that are you have selection versus the physicists would call this, and mathematicians would call it diffusion, but biologists call it drift. So drift diffusion is, what you talk about, say, with, holes and transport in a semiconductor, but this drift here is diffusion. So you see, we run one of these examples again, and where the blue recessive trait is going away, and it does so, but it does so in a noisy way. So nothing really new happens. You might, the population wasn't that large, but if I reduce the selection coefficients and increase the population, I can get the same sort of behavior. So there is sort of a continuum limit to this process. So let's see. Here I ran the program 10 times, and I just pressed run on IPython 10 times, and an unlikely random walk happened where the mutation that was supposed to go away got fixed. And we can play with parameters, but I think the point to take away is that you have this, competition between selection and random walk, and they could be of the same importance, or one could be, more important than the other. Okay. I'm going to sort of end here with a bit of a paradox, or maybe it's not a paradox, but this would be something where there's, asexual reproduction. So there isn't sort of, crossing of genes and recombination and trying different things out. And I'm going to assume that Mueller is the guy who came up with this, and, a ratchet is something like a wrench that only turns one way. So, and here the ratchet is that deleterious mutations are going to pile up. So, oops, I didn't correct some of the notation, but this should be lambda, I think. So I'll correct this before I post these slides. But we have I decided to write this as a continuous process. So n is the number of mutations, and this is always bad to have these deleterious mutations. So you get that this population, the genomes with the most mutations are selected against the fastest. And then you have, this is the lambda is the rate at which new mutations are put on. So you remove something from the population with n mutations when it gets one more, but then you get the population with one less that sort of flows into your n mutation bin, with the same rate. And then we're going to add sort of a constant term. So we're going to assume that the size of the population remains constant. So there's some other population ecology mechanism that maintains the population of a fixed size. So that doesn't float. So if we want to look for a steady state solution, then for the zero case, this term is absent. There isn't anything less than 0, so you have to match these. So alpha is equal to lambda, and you get this recurrence relation. I guess I could have put this on the other side, and you get a sort of Poissonian distribution for the number of mutations you have. So this sort of makes sense that it's the ratio, how fast are the deleterious mutations being added, and that's attenuated by how fast are they being selected against. Okay, so nothing particularly bad happens in the case where the population is infinite, but let's consider sort of finite population size effects. So you're going to have, this is like the measles situation that we discussed earlier. So there's a finite sort of pristine, no mutation population with P0 is non-zero, but it fluctuates. And then at some point, it'll fluctuate to zero. So that's a turn of the ratchet. And then you get a new equilibrium, which is sort of the same, except this n is above the minimum number of mutations. So you see these deleterious mutations just collecting. So this is kind of interesting. It's a somewhat paradoxical result. And, why doesn't this happen? This would sort of mean that everything would die, basically, and there wouldn't be an improvement in fitness. So there are no positive mutations. Mutations never turn back, but it's interesting to think about. One thing that's sort of interesting about, so we think of humans as being a diploid organism, and I mentioned that the sex chromosome is a bit different, and also the mitochondrial DNA. So the mitochondrial DNA is in these organelles. It has its own DNA, and it turns out that only gets inherited from the mother. So you have the sort of same genetics as from a virus. There isn't, this mixing up. And then the male, for the female sex, you would have two X chromosomes. So, and for women, among humans, you have these same mechanisms for mixing genes, but then since the Y chromosome comes from the male, the genetic information just flows from the fathers, which creates a simple linear tree. There isn't this sort of branching out that there are many, 2 parents, 4 grandparents, etc. And there's some amusing papers that talk about, Mueller's ratchet explains why the Y chromosome has so few genes and that it has lost genes over the years. So the withering away of the Y chromosome. It's sort of interesting that with other species, for example, the rooster, it's the male that is homozygous and the female. So that is, it works different ways in different species. So this is an interesting paradox. Okay, so we talked today about mutations and variation, and in some very simple models sort of explored how you have randomness and with the diffusion and also selective effects, which would be pushing in a sort of almost deterministic direction, a response to these random mutations. So here is going to viruses. This is, or the, different corona viruses and rough, and based on the differences in the genome, it's possible to establish, or at least establish with a high likelihood, what are the family trees, or the if these different strains, and this can be done for all sorts of organisms, to do sort of genetic molecular paleontology. So we talked about some very well, maybe not that explicitly, but stochastic models, that you have these you could have a model where mutations just happen, a base pair changes to the other three randomly, and then with that model, what is the most likely link, or most likely tree. So this sort of is an elaboration of the ideas of this, genetic clock, and that mutations are mostly neutral and markers. Now, for this to work, it doesn't really have, there could be lots of mutations that happen that are really deleterious, and they just die off and you just don't see them. So that wouldn't cause a problem. So you don't have to assume that any random change will have no effect, but it's just the changes that you actually observe are more or less more or less random. So that's sort of the central assumption. So we will look at that, and we will also look at, we can ask some questions like, suppose I have a certain protein, and I can find its, homologous genes, or genes from other very distant organisms where, there was a common origin for this gene. Well, if a certain protein and that sequence were really important, like it determined the way it folded, or some essential function, then when you look at all these very different organisms, you'll find that similarity lines up. But then the ones that aren't that important, you see a lot of variation. So looking where there's a variation is a way of seeing what is important and what is noise. And that's sort of one of the kinds of things that you can analyze with bioinformatics. In other words, rather than just taking one genome, the human genome, and studying it to death, you learn a lot about what's important by making comparisons to as many different organisms as possible. So I will stop here for questions. Thank you for your attention.
Okay, Martin, thank you very much for a very nice talk. And let's see, do we have any questions? I don't see any questions in the chat, and I assume the students can just unmute themselves and ask question. And to the best of my understanding, we still have a book voucher competition happening. So please participate. Oh, the students are a little bit shy today. Can mutations disrupt normally gene activity and cause diseases?
I didn't hear the first part of it.
Can mutation disrupt normal gene activity and cause diseases?
Yes. I mean, the sickle cell anemia was one example. So if we, there's sort of something special about that, because if you sort of think of it as that mutation appearing, it would first come up as an advantage in a sort of malaria-rich zone. But then as more and more people acquire this positive trait, having two of those genes will be selected against. So, sickle cell anemia creates all sorts of things, blood clots, lack of red blood cells, and can lead to death. There are also, just defective genes for proteins that are needed will cause disease. I know that in my family, a lot of people have colon cancer. So and because of the incidence is much greater, it's obvious that it is, something genetic. And, that's why when you go to the doctor, often they will ask you about your family going back as far as possible. So those are things to look out for: susceptibility to cancer. So there are a whole host of genetically caused diseases, or diseases where it's not a direct causation, but more of a susceptibility. Yeah, I just gave one example.
Okay, very good. Thank you very much. Any other questions? No, and I just really hope that okay, so that's going to happen in the next lecture, but I really hope that during the next lecture, Martin could convince us all that there's nothing so special about the current coronavirus, and it's not engineered to kill us all by some evil genius, but it's done by kind of, it happens by evolution naturally, and it's kind of one of the possible outcomes of what's to happen and what to wait for. And not only on this genetic tree, unfortunately, but on many others as well. Yes. Okay. Do we have any other questions? Okay. In this case, I assume that we could probably give a prize for the best question to the only question which we had. Refiloe, if I spell your name correctly, can you please share your email in the chat so that Francesco could contact you after the summer school? And if we don't have any more questions, I think we could wrap up this session. I mean, it's sad that you don't have questions because it's such an interesting and topical subject. And in case you are interested, in the afternoon, I will give my fourth lecture, and it will be related to the open quantum systems and possibility of simulation of open quantum systems on the quantum computers, as the future ones, as well as the current ones. Okay, so I still do not see, okay, now I see email. Okay, wonderful. Thank you very much. I think we all could thank Martin again. Thank you very much, Martin.